So Herman Bloch was a Ph.D. student who left here in 1936 and about 81 years before today's speaker. And he was not only a leader in chemistry, chairman of the board of the ACS, one of the directors of Allied Signal. He had 270 patents, and he moved seamlessly between industry and academia. He created conversations, created tools, created opportunities, both inside of the academy and out. And this lecture was generously given to the Department of Chemistry by his family in remembrance of Herman S. Bloch, so that we could honor people who also contribute to the academic knowledge and do so from industry. And so today, John, we have, where'd John go? Come here. We have a medal for you, please. So we have, this is, so he has collected a lot of medals this year. But I will note that we invited him for this lecture long before those folks over there recognized him. And as I was warming up for this, I was looking at the 26 lectures, now including John, and I went to ChatGPT and just asked how many of these folks have won a Nobel Prize. And I'm very proud to say that there are now 3, but when I asked the AI engine, it only told me 2, and it missed one. It gave me Lou Bruce, and it gave me Francis Arnold. I'll leave it to you guys to figure out who number 3 is, but I got to say, this AI thing, I have some reservations. And so with that, I'm going to introduce my colleague, Tobin Sosnick, who will tell you a bit about today's speaker.
Great, thank you. Hi. First, let me thank the Bloch family for supporting this award. Also like to thank you for making me an honorary chemist for a day so that I could give the introduction to John Jumper. So my name is Tobin Sosnick. I'm the chair of biochemistry and molecular biology. John was co-mentored by me and Carl Fried, a member of the chemistry department, and the
I don't know where you're going next.
What? There we go. Yeah, James Franck Institute. Thank you. Anyway, yeah, so this is a phenomenal event here. It takes it back to thinking about all the times and so forth. But so I have the honor and privilege of introducing the 2024 co-winner of the Nobel Prize in chemistry. He won it along with the Miss and David Baker for work on structure prediction and protein design. John specifically worked on AlphaFold 2, as many of you know, and this was an amazing program which has fundamentally changed the way we do biochemical research. It used to be you'd have to do something like crystallography, NMR to get structures, but now you can go to this program and get a structure which has 1 angstrom or less accuracy. And that I never thought was going to be possible. Maybe 1.5, maybe 2. But getting to 1 angstrom was big just because it got you far enough that you could say, this is what the protein looked like, this is where the side chains are, this is what the active site, and it's just fundamentally changed the way people do research. You can hardly go to any talk, seminar without hearing somebody say, oh, I alpha-folded it and got this or that. So we really now have the new word in our lexicon to alpha-fold. And all right, so the impact of this work was obvious from the beginning, and that which explains why the paper in Nature came out in 2020. Nobel Prize was given this year. And I'm trying to think of other examples where the impact was just so outstanding that it was essentially immediately given, in my opinion. Anyway, phenomenal stuff. So Carl Fried and I co-mentored John from around 2011, 2017. It was great times back then. So I often now, when somebody shows up in my office, they find out John was a student of mine, I get the question is, how did John's Ph.D. thesis differ from AlphaFold? And then, in fact, I consider them similar in one sense. We're trying to look at protein structure and dynamics, but quite different in another. AlphaFold 2 is a structure prediction algorithm. It takes a sequence, predicts structure using AI, machine learning, whatever else they could get their hands on. Okay? Sometimes use physics, sometimes throw out the physics. Anyway. So now, because everybody comes in and asks this question, now the second slide in my slide deck is how John's thesis differs than AlphaFold 2. So anyway, so John's thesis was to design an algorithm, molecular dynamics algorithm, which would fold proteins, right? So we have one thing which is sequence to structure. That's important. That's what 90% of the world want. But some of us were interested in actually how it went from unfolded to folded. And that's John's thesis is to develop a molecular dynamics code, which turned out to be 10,000 times faster than normal, but one which was principled, one which was based on physical chemistry principles of hydrophobic burial, hydrogen bonding, backbone entropy. Could we include everything we think is important in protein folding into an algorithm which is able to fold proteins? And we were remarkably successful, or John was remarkably successful. Not as successful as AlphaFold in predicting a structure, but the goals were different. John's goals were to understand the dynamics of the folding process, and for reasons we're not entirely clear, we're at around 80 amino acids are sort of a limit. We'd like to go somewhere bigger. But in my eyes, this is sort of a definitive study of how you include biophysical principles in a molecular dynamics package to fold the protein. And so in that, I'd say John was remarkably successful. We're still using this code. It's called Upside, and I'm trying to remember where we got the name.
So there's two answers. The acronym was using protein structural information for dynamics estimation, but it was really found because I knew I wanted protein structure and dynamics in there, and I searched through the dictionary for everything that has those three letters in order. And I found that and postdoc, and I figured if I named the package postdoc, it would be the least searchable academic package in history.
Okay. Anyway, we still use Upside, anyway, a number of groups are doing it. Aaron Dinner's group uses it, Marco Sotomayor. My group uses it. We're using it to pull one thing, look at dynamics, hydrogen exchange. It's really sort of amazing. All right, so one of the good and the bad things having a student as good as John is getting him to do something you want and he doesn't necessarily want to do. So we have this code, and I occasionally make a suggestion. He said, nah, that wouldn't work. And then I said, what about this? Nah. Then occasionally I came up with one idea, which is use 2 beads per side chain instead of 1. Not important what the details are. I finally convinced him to try something, and sadly it didn't work. All right, so looking back, those were amazing times. You know, some period you have labs which go up and down. In fact, I've been really fortunate here. My lab has been pretty steady, always having couple great people in. But looking back in that period there, that was really phenomenal times. What was, I wouldn't say surprising, is that John got involved in a lot of projects, and he instructed a lot of the other students in the lab. And if you look, one way to look at this is what's the ratio of the number of first author papers a student has to the number of second, third, nth author. And if you look at that, you get an indication of how impactful a student was and how generous they were with your time. And so John's number was 2 first author, but 5 second author. And those he made couple PhD theses possible. This was amazing times, and also I think it was unique times for UChicago, or at least let me rephrase that, as I think we only would have had this sort of times had we been at UChicago. So this was Carl and I, and before that Steve Barry, had worked together for the previous, nearly 20 years, and that was a great time. And I'm not sure that sort of co-mentorship, even though you weren't formally in one of these dual mentored programs, worked. And that was, I think, a reflection of the UChicago one. There's a style people were able to collaborate together. But also, I think it's an indication of the UChicago thinking. And let me just close with a quote from one of John's acknowledgements. I also want to thank my lab mates for vigorous discussion and a willingness to argue forcefully in and out of group meetings. So you can imagine what it was like, both during when you showed up in the lab, which is maybe every week or so, but also just generally when you're hanging out with John. So anyway, John, phenomenal work. I'm really proud of you for having just solved the protein structure prediction problem. So anyway, thank you for visiting, and look forward to hearing your talk.
Hi.
And I'll pause just for a moment to say nice things about Tobin, and especially that I had
I'm
done
listening to a
computational.
Nobel laureate lecture at the University of Chicago, which has just been called to my attention.
Okay. That honestly, one of the great things about being in Tobin and Carl's lab, and they really had a joint lab at that point, but you know, they worked on experiment, they worked on theory, and they worked at the intersection. And I thought it was a great place, and I came out of kind of doing prior computational work saying, yeah, but what do experimentalists really want, right? I know a lot about Boltzmann distributions and not so much about pipettes. And I think just working shoulder to shoulder with experimentalists, sometimes tiring. If I hear one more lab meeting about protein expression and purification, I'll scream. But it's a really important experience. It's really important for computational scientists to do the work that causes people to do new experiments, and I couldn't think of a better place to learn it. Okay, let's talk about AlphaFold. And I'm going to talk a little bit, and I'm going to try and give you a sense, not so much, actually, I'll just figure out who the audience is. And I like to figure out, you know, do I have biologists, chemists, machine learning people? Let's just do a quick, who here knows what tryptophan looks like? Please raise your hand. Excellent. Who here knows what layer norm is? Can write the formula for layer norm? Okay. Good. I think, all right, so I've got a sense of the audience. So most of you know tryptophan, and for that I'll apologize when I give a little bit of a biology intro, but it seems nice to do. And I'm going to talk about, you know, it's really, really obvious why AI needs biology, right? We need a wonderful set of problems that really, really matter. And biology is one of the places to get it. It's incredibly important. But why does biology need us, in my selfish opinion? And I think it's really because biology is this huge horizontal science, that there's a tremendous amount of accidental complexity and just general complexity that comes in an evolved system in something as complex and irreducible as the cell. Right? We have 20,000 genes. We have this vast array of cellular machinery. And there are all sorts of incredible scientists, many in this audience, who shine a very bright spotlight on a very small region of this biological concept space that understand one system or another. And through incredible and painstaking work and someone's AirPod, we find out something about the biological world. But what we need AI is to really generalize this, that at 20,000 genes with many, many interactions, we aren't going to pick out each one. We aren't going to be able to study and understand each one. We eventually need biology to fill in the holes in all the brilliant studies that have been done. And I think we'll see more and more of this. And I think biology is a really great place for this to happen in these like incredibly crowded cellular environments in which we study. Now, proteins, and again, apology, I'll go real fast. But these are, when your DNA is the building blocks for life, one of the big things it's telling you is simply how to build proteins, how to build these tiny nanomachines. And typically, and it's very, very easy, cheap to read DNA. Many of you in the audience probably have had your DNA read. I bet no one in the audience has had their particular proteins structurally determined, right? Reading DNA is, you know, cents, maybe $1. Finding a protein structure, to put it in economic terms, is maybe $100,000, right? Is often a year or two of work of someone who's going to get their Ph.D. for this work. And so really, we have this incredible opportunity where we have this, you know, DNA revolution giving us so much DNA, this being the instructions often for proteins, which fold up spontaneously into ordered 3D structures. And so we're converting something kind of a cheap piece of information into an expensive piece of information, and it's biology which has an element of evolution and an element of physics that helps us to determine the final structure. And as well, I think really to remind ourselves, these are chemistry. These are simply atoms joined together, right? There's only kind of 20. This is actually pretty well set up for a computer in a nice way, in a way that small molecules really aren't and are really unpleasant in a computer. Right? You have a 20-letter alphabet. It fits within the English alphabet. Each one encodes something that appears many, many times in our data sets. So we're in a really nice setting that even though these are molecules, you can think about all the patterns, all the interactions, all the pairs of amino acids that are favorable or disfavorable occur many, many times in our data sets. So it's a very nice problem for transcribing to a computer. I think that's part of the reason, other than the importance, that computational biologists have liked it so well, is you have this compact description of the input. The outputs are really, really exciting, really complex, right? The atomic graph on the left-hand side. And for the non-biologists in the group, no, biologists can't read that atom diagram any better than you can, or not much better, right? So Jane Richardson, and we invented the ribbon diagram that basically traces the backbone through. And, but the structural information that we're getting is also really important because it is about what comes together. It's about the 3D shape. It is about the fact that even though this part of the protein and that part of the protein are very, very far away in the DNA in the linear sequence of bases that encodes the amino acid residues, they're very close in space. And we want to know that fact. And we're simply trying to learn what is close and what is far in this geometric problem. And we have very repeatable data. I'll go ahead and skip this, but proteins do stuff. And, you know, finding this has become important for about 70 years. And it has remained hard for 70 years. We've gotten better at it, but it's remained very, very hard. And I think I find it fascinating that to see something, you know, biological but small, right, smaller than the wavelength of visible light, we go to, this is Diamond Light Source in the UK, if I remember correctly. Those are cars around the edge. Right? Because we want to image something very small, we need very, very bright X-rays, and we have kind of the byproducts of physics to thank for that. And then finally, even a mathematical problem at the end to phase it. And 200,000 protein structures are known. And if we sit back and think about the investment order of $100,000, think about how intimidating that fact is. And these are the publicly known and deposited. That 200,000 times we've done this incredibly hard thing. We've deposited it, and the next one has remained hard. But we've determined billions of protein sequences. Protein sequencing has gone really well. And so you can say that we are learning experimental structure, we're learning experimental sequences at about 3,000 times the rate we're learning experimental structures. Right? Our ignorance grows every year. And in fact, we add about, I think, 15,000 experimental structures each year now. And so this is, even with cryo-EM, this has stayed hard. There's some conservation of trouble. There's nothing that says that protein structure determination couldn't become easy in the future, but it hasn't yet. And so we want computational approaches. We want them from an intellectual point of view, but we also simply want them in a practical point of view. That if you want to study a system, you don't want to wait a year to get the structure of it. And kind of the first place and where my kind of twisty, tangly career started was in simulation. And actually, even before my Ph.D., I ended up, by total chance, at this company doing protein simulation, DE-Shaw research. And one of the things to appreciate when we think about simulation, simulation is the raw expression of the reductionist paradigm. We shall figure out the rules of motion. We shall code them into a computer. We have many atoms, but that's fine. We know how to apply that. And we will do it again and again. Right? We will have our box. There's our protein. There's our water. It's a very kind of seductive picture of how we're going to understand this. And then we just need a potential. But of course, Schrodinger's equation is expensive and annoying. And so we will use molecular mechanics, right? We will use this approximate form. And I love Michael Levitt's slide on it. I love it because it clearly lays out the term, and I love it because it uses Comic Sans. And it helps to remind us, actually, that when we do molecular simulation, we are not using the force field handed down by, you know, God or nature. We're using a approximation, a cartoon approximation of the physics, right? This is non-polarizable. Most of your simulations are non-polarizable, right? Other than it has no three-body bonded terms, etc. It's a approximation that's worked shockingly well, has been tuned over many years to work surprisingly well across a wide range of problems by tuning on very small systems and then applying to very big. But we need to remember it's only approximation. It's probably best thought of as a machine learning model. In fact, you can even talk about pre-training of this on quantum data and fine-tuning on things like helicity of peptides. And this is kind of an approximate thing. And you have a second problem, which is that it's very, very, very hard to figure out what that molecular mechanics force field is trying to tell you, at least in terms of the long time scale distribution. A lot of people would say, and I would remember people would say, I ran a very long MD simulation. How long? 3 microseconds. And I would say, okay, but I don't have any friends that pipette that quickly, right? This is not the time scale of biology. And more importantly, it's so expensive, it's really hard to figure out what works. It's really hard to stack improvements. Because if you want to tell if one method is 5% better than another, you're going to have to run dozens and dozens of MD simulations to validate that change. And so you have a challenge. You have a method with a lot of successes and a lot of impressive results, but also one that's hard to systematically improve because it is so very expensive to use. Even though it feels right and it creates one of the most beautiful things. I absolutely fell in love with molecular dynamics simulations and never ended up leaving biology. But I do remember when I had worked at this company which had custom hardware for molecular simulations, and I thought, I still love these problems, but I don't really want to go here and do, I was doing 20 microseconds of all atom simulation a day, which for the people in the audience is a lot. And I didn't really want to come here and do less simulation in my Ph.D. than I did, Tuesday at my old job. And so joining Tobin's lab, and they had done some kind of really interesting work on how do we try and use PDB statistics in order to make very coarse grain, right? You need to get rid of most of the atoms if you want to go fast. And so my thesis, which was better described by Tobin than I'm about to describe it, but was really an attempt to say, can we rebuild simulation, which I still loved. I still love capturing the entropy of the backbone, et cetera. But can we rebuild a coarse-grained simulation from PDB data? Let's just start entirely from data. Forget quantum data, because we're coarse-graining even further than just to the atoms. And so it was a lot of work in how do we do this? And then going and reaching over and reading a bunch of papers in the machine learning literature, the AI literature. Actually, Jeff Hinton's work was directly related. He invented contrastive divergence together with others. Jeff Hinton having won the Nobel in physics together with Hopfield this year. But how do we take those ideas and transcribe them into molecular dynamics and build a molecular dynamics model? And it was reasonably successful, though it still had the problem that, okay, that graph on the bottom left, I think that's probably ubiquitous. Still took a couple days to run. It was still hard to systematically improve. It's, I think, a very interesting method. It lets you answer some different questions. But I came out of this not sure, should we be pushing this harder or should we do something else? And then I joined DeepMind, which is, very much a place that does a lot of machine learning and reinforcement learning and deep learning. And if I can describe the environment of DeepMind, it's something like being at a machine learning conference every day. Right? Any technique you can ever think of, you can find a world expert down the hall. It's a lot of fun. But they were wanting to do structure prediction. And by the time I'd gotten there, they had been kind of into this evolutionary structure prediction paradigm. And this is one that, wasn't our work. There are several people that contributed. But this is saying that, even though we all want to do this, we want to just say sequence and then physics, and then I guess it's Bloch Lecture, chemistry shall yield structure. We all wanted to do it that way. That approach was very, very hard. It didn't fold large proteins. We could do small proteins. Even that required lots of approximations and kind of well proven that, this didn't work very well. But you could also say the structure influences the evolutionary history. If you think of it causally, right? Oh, dear. The structure, basically, when your mutations, if your mutations break the structure, they will break the function. And so what people have found is, well, sequencing is really easy. I can get thousands of homologs in many cases. And evolutionary structure prediction, some people talk about more specific instances like profiles or coevolution, but you should think of it as just being a Bayesian. Well, if the structure influenced the evolutionary history, then I can look at the evolutionary history and learn something about the structure. And so in AlphaFold 1, the kind of system was, well, we'll take this representation of the evolutionary history, which was, for the specialists in the audience, a Potts model, but really a 2D data derived from it. And, well, we're going to predict the distances in the protein, another piece of 2D data. oh, well, if this is an image of something and that's an image of something, so we'll just grab this machine learning, convolutional neural networks designed for recognizing cat images or general image processing, and we'll just give it protein inputs and protein outputs. And that works some cases surprisingly well. And then when we're done, we have these distances and we do this iterative gradient descent in order to find a structure. But what you should really think of it is, the machine learning was entirely standard. But we were putting protein stuff at the beginning and protein stuff at the end. It worked surprisingly well, I would say, for what it was doing. Sometimes it worked very well, but often it didn't. But this was kind of the original AlphaFold system. And as we kind of thought about and we pushed this and we reached essentially what we thought was a limit, this was kind of in 2018 for how far we could push this. One of the things that kept coming back to is that if you think about what deep learning does, this thing that powers, chatbots and image recognizers and voice synthesis and everything else, you have these set of building blocks, these set of computational gadgets, think of them like a polynomial, right, that help you fit data. But there's nothing about them that is specific to proteins. So they're all very generic. They can learn anything. So everything that it knows about proteins has to be learned from the training data. And that's great if you have really web-scale training data. If you have the entire internet of text, you don't really need to put textual understanding into the structure of the network. But proteins, these data are incredibly precious. And I remember I would give a talk about the AlphaFold work internally at DeepMind, and someone at the end would always raise their hand and say, 200,000 structures is not much. Could you just pay someone to collect a few more? Right? Can we just get some labelers for additional structures? Like, not quite. And really, what we needed to do is figure out, how are we going to build a special machine learning for proteins? How are we going to adapt our machine learning and pull some of the ideas that I learned in Tobin and Carl's lab about how proteins work and elsewhere? And how are we going to put some of those concepts in just the right way into the network and make it more efficient? And if you want an analogy, I imagine many of you in the audience have fit a melting curve. And I imagine used a sigmoid and maybe a sigmoid with sloping baselines. I know Tobin made us fit one in the biophysics class. And you did it because there's a good reason the data should look like, even though it has, 5, 7 parameters, there's a good reason to think the data should have that form. It matches how your data is learned. You could also fit your data with a high-degree polynomial. If you have enough data, it would work just as well, but you probably don't. Right? Having a better functional form lets you learn more, generalize more from the same data. And writ large, the question is, well, how do we write down the right functional form to train via gradient descent for protein structure prediction? And how do we really adapt our methods so that we're no longer fitting with generic things like polynomials and we're now fitting with special things that are well adapted to our problem? And that's kind of the core of the research program of AlphaFold 2. Right, and our goal is simply to end up with, this computational alchemy system to transmute the base metal of protein sequence into the gold of protein structure. And on the right is a totally blind prediction. We didn't know that structure. Blue is our prediction. Green is the experimental structure. This is honestly pretty typical for a small protein in terms of accuracy. Right? So we really can get these details right. As Tobin says, you have to get these details exactly right. And, we have this diagram. I think this is one from our paper. I can't remember and it looks complicated, and if you're a biologist in the audience, it's our revenge for all the molecular biology diagrams of various mechanisms. But much like those diagrams, you shouldn't focus too much on this. There's a lesson inside it. And the lesson is really that we started to think about, and I'll explain to you what these fields are, we started to think about the data we're using, how we're processing, what kind of algorithms we wanted to appear in our network. And this, by the way, is a diagram of, in the middle of the network, the thing that you're normally waiting on is this thing called Evoformer, right? When you run AlphaFold 2, it's by far the slowest part. It's something like 90-95% of the time. And this does the core processing that is really doing the kind of geometric engine of AlphaFold. And the way it was built, and honestly it was built iteratively, and I tell this story afterwards, cleaning up all the wrong blind alleys. But we think about this sequence we have and this representation of the multiple sequence alignments, all the different sequences of this protein. And we provide this as data to the network, not really. One of the nice things about neural networks is you provide them data, and it's kind of like hucking it at your friend and saying, you might find this useful, right? And exactly how you find it useful is to some extent your business is learned over the gradient descent, but we help it in some way. So we start with this kind of input data, and we start with, and you can think of this as a representation of the evolution or the biology, right? This contains the evolutionary data. Similarly, we have this number of residues by number of residues, what we would call the pair data. Because we know, for example, physics has a strong pairwise component. We think a lot in pairs. It seems like a good idea. And as we start, we know very little about the structure of the protein. So in fact, we write in there more or less what we know, which is amino acid identities and the sequence distance, are all that we really put into each cell. Each cell contains a neural vector. But we tell it very, very little at the beginning. But now we have something that kind of semantically represents the evolution or the MSA. We have something that semantically represents the pairs of residues, and we start to say, well, what are some good ideas for what to do? Well, we could process each row of the MSA, which is each protein sequence, using a standard operation called attention, what is at the core of transformers, with one modification. As we process the MSA, we will stick in a place where the pair data comes in. And what it actually does is creates a little mask that says, maybe these two things should talk to each other more, or maybe they should talk to each other less. And sometimes it has a very semantic interpretation. We can find one part in AlphaFold 2 where all the pairs of cysteine residues are communicating among each other, and no points to the biologist in the audience guessing what the algorithm may be doing there. But often we find it just has a conception of what might be the structure at this moment and uses that to process the evolutionary sequences in light of that structural data. So you can think of this as kind of regulating how the sequences are processed. Similarly, we take the sequence information and do essentially a generalized correlation called an outer product, and that, or an outer product mean, and that brings data from the evolution back into the structure. And so we're setting up this real conversation, in one case moving what we think of as geometric or structural data toward the evolution and moving evolutionary data toward the structure. We also process the structure itself for consistency, because remember we have n amino acids, but we have n squared entries in this array. And so we have consistency conditions. And so we basically make a set of connections. In each of these, we don't program what is done exactly. What we do is connect things together. The analogy I like is it's like a good advisor. It says you should go talk to this person. It doesn't give you a script to read. It just sets up the connections and lets you figure it out. In a similar way, we're setting up connections that we think are useful for predicting structure. And we did it very empirically. Every time we make one of these changes, we would find out, did it get more accurate? And then we train it in gradient descent, and it learns how to use those connections. And if it doesn't, then we throw it away and we never mention it again. And there are this and many other changes in which we kind of built this structure into the network. And you can kind of say, well, do you know anything about how AlphaFold works? And I won't say I know everything. But we designed one experiment to try and figure out what AlphaFold is doing inside this network. And so what we did is we trained a little extra piece of AlphaFold for every layer that would try and guess the answer right now. And the analogy I like to use is it's like you're given a test, you have 60 minutes, and 37 minutes in, an air horn sounds, and the teacher says, write down your answer right now. And so you write down your best guess. And if you imagine doing that every minute of the test, not exactly like that, but you would have a pretty good sense of what the student knew at each moment or in what order they solved things. And so this represents, for each of the 192 blocks of a standard AlphaFold prediction, this represents the best guess of the structure. And you can actually see some really interesting things. First of all the way by an early layer, like 15 or so, it has a pretty good sense of the individual domains in this RNA polymerase. All except really, one actually, though, forms very late. If you look at this bottom left gold domain, it takes nearly the whole time of AlphaFold before it figures out the structure. And what we really see here is a process of iterative refinement. That what we think AlphaFold is doing is fairly early from the evolutionary information. It extracts some notion of some things that are close together. It builds off in some domains. But then as it starts to piece things together, it can finally solve parts at the end. It's kind of an iterative solution, not one we programmed in, but how else could it work? And in fact, this gives pretty strong evidence that we would take into AlphaFold 3 that most of the time in the network is spent on refinement, on tuning, on building things on top of other things. And really, we think it's iteratively building structure. So it's not spending a lot of time, I think, thinking about the evolution. And the other thing that was kind of shocking on this and other movies is how dynamical it feels. It feels, there are unphysical moves, but it very much feels like it's operating in space, even though for connoisseurs in the audience, the structure module is not involved in this iteration. It's only the Evoformer that is doing this geometry. And when we were building AlphaFold, the other thing that we kind of said, and actually started, just before we were to enter CASP and demonstrate this, due to COVID, we wanted to make some SARS-CoV-2 structure predictions. There were a couple of proteins that had no templates. We thought we might do the best at predicting it. But we really needed some way to tell experimentalists whether this was useful or where structures were useful. And in fact, the whole story of what we would call confidence measures, or trying to get right whether we're right, we did it, like many things in AlphaFold, 3 different times in 3 different ways. We had done it originally because there's a part in AlphaFold where we retrain on our own predictions and we take only the confident ones, and we use this weird ad hoc procedure that, we describe in the paper only because we didn't change it at that point. But then we started to realize, actually, there's a more principled way to think about, when should biologists trust us? Predicting the structure, we're getting pretty good at that. Why don't we just predict our error as well? And the analogy I like to use, again, a kind of test analogy, is you've all come out of a test and a friend has turned to you and said, how'd you do? Right? And you say, well, I'm sure I got question 1, not so sure on question 2, and question 3 I wasn't even close. And you say that without knowing the answer to question 2 or question 3 for sure. Right? You reflect on whether your answer makes sense, whether your process makes sense. Right? So we're able to kind of self-predict, and actually, as we predict, right, the structure, we have the model predict an error. And the way this really works is that training, we can measure the error. And then we can measure, as we predict. So these are outputs that are available from AlphaFold. We measure the error when we're training, right? We know the answer because we always train on something that's in PDB. We have to know the answer. And then we have an error, and we can measure the error of our predicted error. And so it's very simple. So PLDDT is named that because there's an error, really accuracy metric in the field called LDDT. So we predict LDDT, so PLDDT. And then you can do this at training. And if you think about it, this side never needed the actual right answer. And so that's all we do. It's a simple method of getting there. Have been all these complicated methods to assess structures. But what turns out to work really well is just, have the model. It's an expert in protein structure, predict its own error. And the PAE is very similar except for a pairwise notion of error. And works shockingly well. Now, in AlphaFold, we do this, we do many other things, and there's a part I always like to say, or people always want to say, yeah, but what's the one thing that mattered? And even today, I tell you a bit of hero story. But then you could take all your ideas at the end of machine learning, and it's, models are expensive to train, but not galactically so, and you can do knockout on your own ideas. And actually, one is kind of expensive, the self-distillation, so we knocked that in. But the baseline is here, and you can see all these different ideas, each of which I can tell you a hero story about, each of which is great fun. And you can see the exact effect. These are 3 different replicates of training a model without this, of knocking out that idea. And AlphaFold 3 is about, there's 2 ways to look at it, but it's about 30 GDT better than AlphaFold 2 on this metric. So the delta to previous methods is way over here. And you can see each of those ideas I can tell you a hero story about is worth something, but only a little. And this is pretty typical of machine learning. We tell hero stories, but we shouldn't. A lot of it is about the small and mid-scale ideas within it, some of which are very clever, some of which are kind of dumb. Now, when you start doing double knockouts of ideas, removing 2 ideas at the same time, we see some larger effects. Right? I think we solved many problems twice. But in general, all of it mattered. And and this is why also it's such an iterative process. And this is why it's so important to be able to test your machine learning system really, really fast so that you can stack the 10, 20, 30 ideas that you need to come up with a really effective system. And I can say for that 30 GDT, I think our largest single change that anyone made was worth 2.5. Right? And most ideas were worth less than 1, kind of in deltas of systems. So really, piece by piece, inch by inch, we ground our way to something that really, really mattered. And this is very, very common in machine learning. So we should walk away from these hero stories and instead tell hero stories of small ideas, of process that get us to great systems. Okay, you should, oh, that's a terrible slide color. You should, not trust me. I mean, all right, now everyone uses it, but before everyone uses it, since 1994, and you can see John Malt and Christoph Fidelis have given talks on this, but since 1994, people have been saying protein structure prediction is solved. I can do it within 1 angstrom. And in 1994, there were quite a few of these kind of relatively strong claims that we can see parallel to some of the claims, I think, that are made today. And John and Christoph had a great idea. They said, well, if this problem is already solved, you wouldn't mind if we do a little experiment. You see, I have some friends that have solved some crystal structures and haven't published them yet. You wouldn't mind making predictions. And of course, in 1994, they were horrified at the results. They had all engaged in self-deception. And they kept running this experiment every 2 years. And they had the right also attitude to it. It was basically, we're going to give you a sequence 3 weeks later or 2 weeks later. We're going to ask you for the structure. You can do basically anything you want in that time, maybe other than a pipette, but you can't pipette that fast. And you give me an answer. I don't care how you do it. I just want to critically assess how well you do it. And pretty much all the structure labs came together to enter this. And, we did very, very well. This was CASP 14, so they continued this for, every 2 years. Now it's on 16, so 32 years of doing this, real commitment. And we were, 1/3 the error of others in this assessment. It was dramatic. And I think also this result was incredibly important to me. I mean, before CASP, we were pretty sure that unless we had royally screwed up our evaluation, that protein structure prediction was solved. But what we weren't sure of, or I wasn't sure of, is did everyone find the same thing? Was it in the air? Were we all sitting and thinking, in this CASP, I will show everyone that protein structure prediction is solved? And I could imagine something in which 4 different groups announced this at the same time. But no, that wasn't the case. That it was just us that had found the set of ideas that worked. AlphaFold works. Predictions are out. I think it's also important, and maybe not news for many of the biologists, but maybe for some of the students, how does this really work? How does the computational work actually feed into someone's biology? And this is a lovely paper in Science on this protein midnolin that's involved in non-ubiquitin-mediated decay. And this was a protein that came up in one of their molecular biology screens, knew almost nothing. It had been almost completely unstudied as a protein. And they ran it through AlphaFold, and they get this kind of weird thing, including 2 things that I'm pretty sure they didn't name catch domains until they did later experiments. But it has some stuff in it. How do you figure out, how does this work? They know that there are lots of genes whose expression is knocked down when this protein is upregulated, but how does it really work? And they did an experiment, and they had about 500 genes that their high-throughput assay suggested were somehow regulated by midnolin. And they ran all of them through AlphaFold multimer. And what they found again and again was the same motif, that in fact 2 domains of this protein would wrap itself around a strand on the other protein. Something like 40% predicts almost identical structures for different targets. And, of course, they're being real and proper scientists, also went and validated it, not by solving, 200 different or 100 different protein structures, or 200 different protein structures, but by just going and knocking out this region and finding that for each protein, when they knocked that out, the degradation was abrogated. And that pretty much confirmed the mechanism. Actually, I think there was one, and maybe it was FOSB, I can't remember, where they found it actually only partially reduced the degradation. But AlphaFold actually had predicted 2 different binding sites for midnolin on this protein, and then knocking out the other one actually fully removed the degradation. But I think it's about how this computational prediction, I love it because it is how does this prediction enable molecular biologists to take the next step in their story and to use it to inform their next series of experiments, to confirm something, to propose a mechanism. And I like to think without AlphaFold, they certainly would have published these great results on midnolin as a suggestion of what this does, but they wouldn't have the molecular mechanism. And the fact that we got this all in one paper instead of a couple of years later in other papers after the structure is solved, I think is an indication of how these computational tools enter as a piece of experimentalist puzzle. And, I think of the highest goal of computational scientists is really to convince an experimentalist that they should do a different experiment, a better experiment, because of what they saw. And for the computational audience, really think about who is going to use your tool and who is going to choose to order a different mutant because of what they see from your computational tool. Now, this was all the AlphaFold 2, wonderful work. There's AlphaFold 3. And AlphaFold 3 was really sitting around and saying, well, AlphaFold 2 is great and all, but kind of made for uncomfortable talks, because at the end of the talk, someone would say, beautiful, wonderful talk. Maybe I'm exaggerating. But they would say, beautiful, wonderful talk. However, my protein is phosphorylated, and I just want to know what it does when it's phosphorylated. What's different? Someone else would say, beautiful, wonderful talk, but can you tell me how it binds DNA? And I would get all these, and I would have to say, for all of these questions, really, well, I'm afraid you can't even ask that question of AlphaFold, right? That's not for AlphaFold 2. You can't even describe it. It takes, the 20 standard amino acids. And it was really, we kind of said, well, we ought to be able to do more. How far can we push these ideas? And we started to call this goal whole PDB. If the atom's in the PDB, we ought to be able to predict it. And, of course, I'm telling you about this because it worked reasonably well. But what we really did is started to adapt the methodology. And we knew that we would have to do two things. We'd have to make it more accurate and kind of continue to grind on machine learning accuracy. But the second was come up with some new ideas about how to do it. And honestly, there's another thing. People said, and they were not wrong, that there's much, much less data for all of these other types of interactions than there are for protein-protein interactions. And so there's a real and open question of how far can you push it. There's no question that we would do worse, but would it be useful? And it was really an exercise in how far can we push it for small molecules, for everything else. And the answer was reasonably far, better than, for example, docking at protein ligand prediction. I think another big surprise, and one that had come out in AlphaFold 2, is AlphaFold 2 is kind of complete rubbish for antibodies. For antibody-antigen binding. And people said, well, there's no coevolution, right? We'll need to do a whole new, completely different type of thing to do antibodies. And that was one reasonable hypothesis at the time. It didn't really sit right with me, but there was another question of, well, maybe antibodies are just hard. And so what we found is that we did basically anything we tried to do antibody-specific work, fine-tune only on antibodies, all that stuff didn't work at all. And what worked in AlphaFold 3 was really machine learn harder, right? And really drive the accuracy on protein-protein interactions. And you can see this and you can say, and this only goes up a little bit, especially remember this is the top, on all protein-protein interaction. But protein-antibody interaction goes up quite a bit. And then this is a really general pattern in machine learning where some cases are harder than others. And the right answer is not often to do something fundamentally different, but simply to get those extra few points of accuracy because they're often dramatic on hard cases. As one of the surprises of AlphaFold 3, and actually I can tell you where in AlphaFold 3 it happened that this worked that well. And when we were thinking about making AlphaFold 3 and how do we do this, coming back to this thing I showed you before and really thinking, well, most of this time is spent doing geometric stuff. Why did I show you that Evoformer graph, which I believed at the time we made it, that showed us processing the evolutionary data the whole time when most of the work here looks more structural, looks more geometric? And so we said, well, instead of 48 layers of processing evolution, we'll do 4 layers of processing evolution, and the rest we'll do a simpler block called the paraformer, which essentially just removes an evolutionary track and otherwise does the same thing. And this was really inspired by looking into the network, analyzing the network, coming up with a hypothesis and testing it by changing the structure of the network. And then we kept it because we measured and it's more accurate. And it actually turns out this particular change, which was made before any of the others for the specialists in the audience, was really the one that gave us better antibody-antigen. I think it started to stabilize the training. But just these kind of simple and inspired changes and thinking about the neural network itself as an object of study that we can in fact improve led us to make improvements. The other big thing we wanted to do especially was handle general ligands, right? Things not derived from residues. And let me just confirm, And the problem with general organic ligands is that both nature and organic chemists are clever and they make complicated things. And they don't have simple repeating units. They have some motifs that repeat. but how are we going to treat these? And when we were thinking about it, we really started to say, well, we've got two paths. We can do all this kind of custom crafting that we did for residues, and we can try and generalize it to ligands, and we can do RD kit-like things and think about which bonds are rotatable, and think about all that other stuff. Or we can get really lazy, and we are really lazy and industrious, and we can just go to an atom-based representation for the ligands, and we can try and make that work. And for the ligands and the post-translational modifications, we said, well, why don't we try and rebuild it around atoms, and then we can see if we get ligands for free. And so we went to what's called diffusion-based structure prediction, which other groups had been experimenting in related, I think, somewhat different ways. And here this is, it's a little bit technical, I may skip some of this, but basically it's about making repeated predictions. So now instead of a very geometric processing, we have a very non-geometric processing. For the machine learners in the audience, the features are all raw Cartesian coordinates. It's like the dumbest representation you can imagine, but we do an iterative prediction that iteratively refines in this diffusion approach that I won't go into, and that lets us learn features on all scales. And pretty immediately it worked about as well for proteins, but then we add ligands and it works pretty much the first try. Actually, I think we were doing nucleic acids first, but it really was, switching our representation, and then we could just huck in ligands like it was nothing. It was just extra data. Now, the dark side of doing this that you've all probably experienced is AlphaFold 2 had a very lovely feature that its wrong answers look stupid. More specifically, and this actually led to some panic in AlphaFold 2 when we were first making it. We made it, we did well in CASP. We're like, okay, let's make a bunch of predictions for random proteins. And so we'd grab a sequence from UniProt or from the human proteome, and we'd throw it in there and we'd get a spaghetti monster. And we all panicked and we said, oh no, AlphaFold 2 is not good at structure, right? Or it's only good at the kind of things that appear in PDB. Until Catherine looked up in UniProt the disorder annotation and says, by the way, all the stuff that has no structure is also disorder annotated in UniProt for these proteins. And it really became an empirical thing that, there's a lot of disorder in proteins, which is not news to molecular biologists, but structural biologists don't like to look at it. And AlphaFold really put that in your face with a nice long loopy thing, became ribbons. And people would say it was actually very interesting to see the sociology of this when watching Twitter, when someone would say, AlphaFold is complete trash. I put my transcription factor in, and it didn't give me any structure in the tail. And someone else would say, that's maybe kind of right. But this was very good. Now, AlphaFold 3 sometimes would do the good thing, but sometimes actually the first time we trained it would often produce the thing on the right because of this diffusion, which is a slightly different generative model, would mark regions as low confidence, but would also give them a horrendous structure. A, it was distracting, but B, if people didn't look at the color, and I'm really glad that this came for AlphaFold 3 after people had been trained to look at confidence, you would think it doesn't know anything. And so we try and we get some success in training AlphaFold 3 also on AlphaFold 2 structures at a certain percentage just to induce this long wavy behavior. It doesn't tend to work when DNA or other things are present. There's some technical things I think it could be improved. But we really, if you think about it, this diffusion was a change, and I haven't gone into it because of reasons of time, but it gives us one set of advantages, it gives us another set of disadvantages. We fight those disadvantages. We have to engineer. We have to think about it. We have to put in our knowledge of biology, our knowledge of machine learning in order to make this work. And I think I want to step back kind of at the end and say, this will be done again in many cases. Many of you in the audience are working on machine learning. And I like to think of this as an amplifier of the work of experimentalists, that experimentalists get these wonderful pieces of data for the systems that they're interested in, and tools like AlphaFold and general machine learning help us complete this picture. And I hope and expect that we will do it again and again, sometimes repeating, sometimes doing special experiments to collect the data, but I think often, even on found data, it's proven incredibly powerful. The language models we've got have been found data of internet text as well. So I think we should see this pattern repeat many, many times, and I hope we'll be able to drive it forward. I want to thank, many people. AlphaFold is the work of a team that I've been privileged to lead. The core team is about 15 people. I'll actually show a picture on the next slide. But we've benefited from, a lot of advice, a lot of interesting interactions with people all over DeepMind and outside. AlphaFold database is only possible because of our collaborators at EBI, both on a technical side, but more importantly on a biological side, giving really great advice. I think Samir Blanker, when we saw the kind of unstructured regions, I wanted to, hide them in the AlphaFold database, and he says, no, you should put them right in front of people, both to remind them that this is a prediction, because no molecular biologist will think this is a real structure, but also to remind people these disordered regions exist and we should think about them. The CASP community, who has measured so well, and especially PDB and the experimental community that gave the data we were able to train on, including, I imagine, many people in this room. And with that, I finally want to say, AlphaFold is the work of a team. They're a wonderful group of people, and it's great to be able to kind of celebrate this accomplishment that was done by some really, really great friends. Thank you.